Thank you for the introduction, and thanks to the chairs of this conference for inviting me. So today we're going to talk a little bit on how we can really put together biological neuronal cells, so living neurons, with artificial ones made of organic semiconductors. This is mainly because in my lab, we're trying to engineer materials and devices and platforms which can be integrated and communicate with the nervous system, so ultimately, specifically with the brain. And in particular, we go from material design synthesis to chip integration that goes also towards implantable probes for a number of applications. So the way we do this is really going through the pipeline from the material engineering, integrating them in vitro platforms, and first validating the interface between living cells and these electrodes or devices like transistors. In a way, we're interested in understanding the communication, so potentially the bidirectional communication between micro devices and neuronal cells, in particular aiming to record the electrical activity, so action potentials, but also impart stimuli, like for instance, electrical or optoelectronic stimulus. And this is, of course, for understanding the way information are processed within living neurons, to potentially monitor also treatments, for instance, in drug discovery or drug administration for example, when pathological conditions arise in living neuronal networks, as well as ultimately repair pathological condition by restoring certain functions in neuronal networks which have been lost. If you think, for example, of like neurodegenerative pathologies which really break certain connections within living neuronal cells. And so the way, when we try to couple together those micro devices as transistors and individual cells, this is a kind of a typical snapshot that you can see. So this is a scanning electron micrograph of a cortical neuron which has been grown on a silicon-based transistor. So as much as this interface looks very beautiful, so the neuron can really grow and nicely engage and adhere on those devices, there are some major challenges that we still need to tackle to improve not only the topological interaction, so the mechanical interaction and coupling between these two very diverse entities, but also we should make an effort in trying to make and go beyond just conventional planar electrodes and devices to make a more functional electrical communication. So also trying to implement, as you know, computing capabilities here that could better than resemble and then also interact actively and adapt with living neuronal cells. So the way we attack this topic in my lab is really into two ways. So our paradigm goes along with we want to build and make electronics that looks like a neuron and acts like a neuron. Particularly, we work on the material design and trying really to reshape materials to resemble structures and morphologies of neuronal cells. For a matter of time today, I'm going to more focus on how we can work on the functional kind of interaction. So I will skip this part, but I'm happy to talk about this offline afterwards. But I would rather tell you a little bit of a couple of stories on how we can really resemble, especially tuning the properties of neuromorphic devices to organic semiconductors to resemble, for instance, synaptic plasticity, learning, and connectivity to ultimately interface living neurons. So, of course, the best way to approach this is really to look at neuronal functions, specifically looking at the synapse structure. So this is where we try to approach the problem. So where the action potential can and are effectively transmitted one cell to the other, and then obviously propagated through a large neuronal network. And so, of course, the main point that we try to look at is really to resemble synaptic plasticity, of course, considering and in general action potential are generated from a first neuron, what we call a presynaptic neuron, and then through the release of neurotransmitters in this synaptic cleft, receptors at the second neuron standing at the plasma membrane of it are actually being activated depending on, for instance, how much neurotransmitters are being released over time. So this is a kind of interaction and engagement that can happen at two different scales. So when it's just in the terms of milliseconds, it's fairly quick. It's called short-term plasticity, while if this type of interaction goes on for much longer time and even more of those receptors are being activated, then we can consider this as a sort of a long-term plasticity. So why using conductive polymers to resemble this? So conductive polymers are, I mean, now not emerging materials anymore, but now very established materials in the neuroelectronics and the bioelectronics field in general. So of course, if we ultimately aim to a direct interface in between electronic devices and living cells, those materials are extremely appealing because they are organic, so they are biocompatible. They are polymers, so they can be easily integrated, and we can take advantage of polymer-based processes to integrate them into electronic devices. But of course, a very important and peculiar feature that they have is their way of interacting with ions and electrons. So they act as intrinsic transducers because they have a mixed ionic electronic conduction mechanism. Last but not least, we can also engineer these materials to better resemble the stiffness and actually the softness of biological matter like the brain. And so we can really achieve low Young moduli when we, for instance, integrate them in hydrogel-like materials. So in our case, we focus on using conductive polymers, in particular PEDOT-PSS, integrated as a channel of an organic electrochemical transistor. So this is the typical structure. So this is a 3-terminal device with a source and a drain and a PEDOT-PSS channel between the two. And then in this configuration, we have a gate electrode standing then in contact with the electrolyte, then also connects on the other side the conductive polymer channel. And so what is very important here is that electrolytes contains ions, and base, of course, in the case of PEDOT-PSS is a p-type material. We can then trigger the movement and the migration of those ions from the electrolyte when we apply a bias at the gate. And let's say based on how many ions are then penetrating into the transistor channel, an electronic current is then generated and modulating over time, let's say, also depending on the transconductance of the channel itself. So this is then a typical, let's say, way of our PEDOT-PSS transistor works. So this is working in depletion mode, meaning that when we do not apply, so when the bias at the gate is 0, the transistor is on, and so we can modulate the channel current by then applying and so decreasing the conductivity, so dedoping the polymer at the channel when we then modulate the bias as an input. And so what is also very important here is that, as I said, when it's 0 at the gate, we have a channel, a current flowing in the channel. And then this is the typical, let's say, behavior, which very strongly depends on the capacitive behavior of the conductive polymer itself, which can be represented as an RC circuit, and the rise in time of the response time of this transistor channel strongly depends on the geometrical factors, so length and width, for instance, or thickness of the PEDOT channel, that also affects then how this capacitor is charging and discharging. So we can also look at this transistor has a synapse, and I'll guide you through this, let's say, side-by-side kind of a snapshot. So on one side, we have the transistor in the architecture and the arrangement that I showed you before. And so we can see the gate electrode as a presynaptic terminal, so the first presynaptic neuron in a synaptic connection. And then the channel, so the PSS channel, as a post-synaptic terminal. Now, when an action potential is generated and comes in the first neuron, this can be seen, for instance, as a train of pulses that are being applied at the gate of the OECT on the other side. And what is interesting here is that we can really mirror then the neurotransmitter release and the effect of a neurotransmitter release in a living neuron as the ionic migration from the electrolytes towards the PSS channel, which of course results in an output current, so in a modulation through, for instance, the conductance of the PSS, which in our case now can represent the synaptic weight of this synapse. And of course, this can be seen as the action potential is then generated in the second neuron. This is kind of what happens if we think of the synapse and the response and the mirror to a single action potential. However, what is also very relevant is to understand what is the response of this artificial neuron when we apply subsequent, so a train of pulses, so a train of action potential as an input at the gate electrode. And so what happened here is that we can really mimic this burst kind of activity and spiking activity at the gate by applying subsequent pulses at the gate. And this would allow for cations, in the case of the PSS, to move into the channel transistor. But then if there is enough time in between the two pulses, to those cations to diffuse back into the electrolyte. In this way, we can really build up sort of a short-term kind of depression, because this process is, as I said, equilibrium is reached, it's fully reversible, and so this can really help us in building a so-called volatile memory. So, of course, another point that we can work on is trying to go from a short-term depression or volatile memory to a non-reversible mechanism. And the way we did that, especially also aiming to have this type of short and long-term plasticity when we couple living neurons in contact with this PSS neuromorphic device. And so we exploited the fact that cells release neurotransmitters. Specifically, we focused on catecholamines, for instance, dopamine or serotonin, because when those kind of molecules oxidize, and you can, for instance, do cyclic voltammetry to identify what is the specific and characteristic oxidation potential of those neurotransmitters, they do something very interesting, because they release 2 electrons and 2 protons. And this additional add-on of protons that is released in the electrolyte can be used to further trigger and modulate the synaptic weight, the transconductance of the transistor. And this is actually what we did. So we had living neurons now directly in contact. Just for optimizing the interface, we use a microfluidic device together with the organic electrochemical transistor. And so what we see in here is just oxidizing the dopamine, as I said. We could, applying, let's say, as an input at the gate, a train of pulses with an amplitude which would match the oxidation potential of the neurotransmitter, meaning that we would first move the cations present in the electrolyte, as I said before, just because of the gate being applied, but because of this specific amplitude, we would further have this add-on of protons, which would then wouldn't allow for the, let's say, full recovery of the doping state of the PSS material, meaning that we would have these conductive states being retained and modulated over time, also depending on the concentration of dopamine being released by cells, which were also happily living on top of those devices, even while acting and doing this type of operation, because as what you see here in green means that cells are alive and active. Of course, we have not only one neurotransmitter in synapses, so we try to step by step increase a level of complexity in this biohybrid connection. And so the next step was to add a second neurotransmitter. So in this case, we used both dopamine and serotonin, but also we wanted to exploit the electrochromic properties of P.PSS. This means that the doping and the de-doping level of this conductive polymer can be both monitored electrically and optically, and also this type of, let's say, conditioning and extinction kind of process within the electrochemical synapse. And this is actually what we did. So we coupled then the electrical system with the spectrometer fiber to do this simultaneous monitoring. And we found out that also depending on the concentration of the neurotransmitter, so serotonin and now and dopamine together, we can really have, let's say, a very nice match in percentage of the conductance level change monitored by the two means of, let's say, of electrical and optical interaction. And we also exploited that to kind of create potentiated synapses, in this case, working together with the oxidative states of these neurotransmitters as individual ones, but also to induce low and high stimulation levels by tuning the concentration when we put them together in the microfluidic system. Of course, with the ultimate goal to interface those devices with the brain, we also worked on tuning then the properties of going beyond the liquid electrolytes, but work on gel-like electrolytes. And in this case, we worked with glycerol, which is an alcohol, and glycerol and agarose. And let's say, just to give you an idea, agarose could be also used in your kitchen, so you can actually resemble brain-like jelly thing also at home. But what is very interesting here is that the diffusive process now of the neurotransmitter strongly depends on the viscosity and also the intercalation of water within this electrolyte. And so from our finding, let's say glycerol was actually a no-go, because the more we would increase the concentration of glycerol in our electrolyte, the more, let's say, barrier and absolutely no diffusion process we could monitor, while in the case of agarose, which is really resembling the brain structure and composition, we had a very similar behavior when we compare, let's say, liquid electrolyte, meaning that we can really have a sort of a liquid interface in between also these gel electrolytes, which is enough to ensure the response and the modulation of the transistor. And this is effectively also what we validated using, again, dopamine and serotonin. And we found also that the neurotransmitter, which has, let's say, a higher diffusivity constant, and so can better also diffuse and modulate the synaptic weight of our, let's say, tissue-like neuromorphic device. Another point where we can really put this type of interface is not only to directly interface the brain, of course, but also act as a functional and more optimal interface also for prosthetics. And this is where we're trying to use this artificial neuron from one side, still facing and interacting with the neuronal cells, but also, for instance, be connected to a robotic arm. And so in this case, of course, we use dopamine to, as I said, potentiate the synapse, but we also wanted to induce a reversible process, and that's where we used hydrogen peroxide to further modulate and recover the oxidative state of the P.PSS channel. And so in this case, we can really now compile together a number of conductive states to, for instance, achieve and perform certain tasks. And with the robotic arm, our goal was to make it opening and closing, and also learning how different objects were, let's say, looking at, and they could recognize them. And this is actually what we did. So we implement then a feedback loop control with a PID system. And so we actually set two targets, like open and close end, based on by setting the current of the as a goal in the channel current of the transistor. And with this, like the artificial neuron could reach those targets by, let's say, alternating the neurotransmitter and the hydrogen peroxide. And this is actually then also later on how we could perform reinforcement learning by, for instance, also integrating a pressure sensor at the fingertip of this robotic hand, and then exposing the robotic hand to, for instance, a tennis ball, so a large object. Impart, let's say, a punishment signal until, let's say, the hand is not grasping the object correctly. And then as soon as the pressure sensor would give, let's say, the effective attachment to the ball, have a reward signal being applied there. And then we removed the tennis ball and we exposed to a ping pong ball, and we started to do a similar concept. So until the pressure sensor would give us, let's say, green light that the pressure applied was such that the grasping was correct, we would impart a punishment signal until we got then a reward one. And then we exposed the hand again to the ball, which was the largest one, so the tennis. And then directly the ball, just in one go, would completely, through the pressure sensor, recognize that and grasp it correctly. This is how, let's say, we are not a robotic group, and so our hand is fairly, let's say, rudimental, is a 3D printed, but nevertheless, this is how this is connected to our control system, and the pressure sensor stands just at the fingertip there. I'll take the last 5 minutes to also mention how we could use those electrochemical transistors to resemble another type of neuron. So what you have seen before kind of resembles typical like a central system neurons, so like from the brain. Another type of integration that we wanted to make was also to resemble certain functionalities which are closer to retina-like computing. So we had to integrate some light-sensitive materials into our transistor. And this is what we did actually. So same structure that we had before. So it's a planar P-dot-based transistor, while we put at the gate a P-dot that is modified through click chemistry with a photosensitive molecule, which is an azobenzene. Azobenzene are very beautiful molecules. We can also make them as a film. In this case, what is interesting is that when you switch on a UV light, you can change the conformation of those molecules from a cis to a trans conformation. And while we were doing this, this would result in a modulation of the gate current. And so now, by applying together, for instance, these pulsing inputs as action potential together with the light irradiation, we could trigger a certain, let's say, conductance level and retention within now the optoelectrochemical transistor over long time. And this actually can be exploited by, for example, let's say, create different type of writing and erasing states, as you can see here. For example, by coupling together this type of input stimulus, so the light and the electricity together, or decoupling them and trying to, let's say, achieve, for instance, writing when we had the light on, and erasing, for instance, when we applied, like here, a negative bias applied at the gate and the source and the drain. What is very interesting, especially in the case when we write things with the light, is that, or when we erase, that this is light intensity dependent, and also the erasing process depends on the amplitude that you're applying at the gate. So I hope I convince, or at least give you an insight that in principle organic neuromorphic devices can resemble the properties of neuronal cells, but they can be ideally also coupled directly in contact with living metals, like for instance living neuronal cells. We can exploit a mean of communication that is already present in living synapses, like neurotransmitters, to make then a biohybrid communication, as well as also take advantage of the properties of conductive polymers to further engineer and modify them, for instance with light-sensitive molecules, to create other different building blocks to then resemble different properties of, let's say, the different neurons that we have in the nervous system.